morning, everyone. We from Group 6 will discuss about biocontrol of plant pathogen. Professors, we will introduce the member of group which consists of my name is Dewi Istikomah with the number 137, Dena Aisyah Puspitasari Wardani with the number 138, Rinta Sofia Nurrahmah with the number 145, Muhammad Ari Setyo Nugroho with the number 149. Next. Formulation of the problem that we will discuss, namely, first, what is a biological control agency? Second, how to increase the quality of biological agents for the control of plant pathogens? And last, how to increase plant resistance to pathogenic hosts. What is biological control agency? Biological control agents, namely any organism which includes subspecies, species, varieties, all types of protozoa, insect, bacteria, fungi, virus, which in their development stage can be used for the purpose of controlling pests and disease or organism. Next. Improving the quality of biological agencies using molecular biology technique. Technique 1, genetic improvement. Genetic improvement of biological control agent is very important in biological control of plant pathogenic through genetic manipulation by producing super and superior strain according to needs.
One, genetic improvement. Research on genetic improvement of biological control agents have been widely conducted, especially Pseudomonas fluorescens, the first report on the cloning of an antibiotic gene by Gutterson et al. Antibiotic DAPG is an antibiotic produced by many Pseudomonas fluorescens strains which has the activity of suppressing the attack of cell-borne pathogens. 6.5 kb fragments of genomic DNA that can be transferred to the Pseudomonas fluorescens, receiving strains that cannot produce DAPG, resulting in strain that can produce DAPG. Improving the quality of biological agency using molecular biology techniques. Number 2, protoplast fusion. The integration of intraspecific protoplast of Streptomyces melanospora fashion AF76, a gildanamycin producing biological control agent, resulted into recombinant strain which had different antagonistic abilities in inhibiting the attack of the pathogen Bacillus cereus ATCC 14579, Streptomyces cabeus AF35, and Phytophthora fragariae variety Ruby 390. Number 2, protoplast fusion. Where the recombinant strain FP454 produced the antibiotic gildanamycin and has the same antagonistic ability as the parent strain AF76 by inhibiting the three pathogens above.
Okay, protoplast unification can be done by two ways. The way one is chemically with polyethylene glycol or PAG. Protoplast can occur with PAG at a pH lower than 7 in the presence of 0.01 mol CaCl2, but at a high pH 9 with 0.6 mol CaCl2. Next. The way 2 is physically using electrofusion. This technique is done by the protoplast from the original source are mixed and given high frequency electric which cause fusion between the protoplast and form a kind of protoplast grains. Next. Increased resistance of plants to pathogenic hosts. The point 1 is induction resistance. Induction of resistance is a phenomenon from the host plant to defend itself from pathogen. Be expressed by increasing defense response such as cell wall leasing causing enzymes such as chitinase and 1,3-beta-glucanase, phytoalexin synthesis, wall strengthening, and cheltrope increase peroxidase activity. For the example, Fusarium oxysporonum pathogen strain can control the incidence of Fusarium wilt in tomato plants. Trope increase activity of chitinase beta-1,3-glucanase and beta-1,4-glucosate. The strength in the cell wall can make it difficult for plant to be penetrated by the pathogen and suppress the spread of intracellular pathogen.
Okay, and next, deposition of cause materials such as lignin or substance may also occur after injury approach, but during a defensive reaction against pathogen attack. The reaction is usually increases, especially around the tissue penetrated by the pathogen. Shimura in 1979 reported by the local infection by Tobago mosaic virus or TMV in Tobago. When can increase calcium deposit when we can plasmodemata, thereby inhibiting to the movement of virus from cell to cell. And next. Okay, this is increased resistance of hypovirulent. Hypovirulent pathogenic strains are also very effective in increases to introduce or resistant into plants to good plant diseases caused by pathogenics in the same genus, such as a pathogen Fusarium oxyperiumon introduced to tomato plants, and to become resistant to Fusarium disease caused by Fusarium oxyperum, and hypovirulent or non-pathogenic strain in the same species of genera usually have almost the same characteristic or the virus strains. So competition for seeds or infection congelation cannot be avoided. This ability causes some of the who are hypovirulent to have the potential to win the competition, thereby reducing disease attack. Okay, and conclusion, improving the quality of your legal against throwing pathogen be done by utilizing monoclonal biology technique, including by the way genetic improvement, the integration of protoplast, and will the increased loss, namely by introduction of resistant and hypovirulent. Okay, this is a presentation of growth seed. Thank you for attention, and thank you very much for your attention, and wassalamualaikum warahmatullahi wabarakatuh.